Welcome back, everyone. This is Dr. Jared Blinch from 2025, and today we're going to start a new section on vision. Today we'll focus on the neuroanatomy of vision and also the conscious and subconscious perception of vision. So vision is important, and if you think of the expression, look before you leap, would be very apt for these penguins here. And vision is often one of our dominant senses. And when we have all senses available, if we compare, say, vision to proprioception, so I can see my hand, right, and I can feel my hand, but vision tends to be treated as more dominant or given more priority because our visual acuity is typically higher than our perceptual acuity. So where I feel my hand is not as specific as visual information. So vision tends to be one of our dominant senses. I did want to add, though, at this point, before we talk a lot about vision, is that vision is not vital. You don't have to have vision to be able to survive and not even to thrive. And three examples of that are Louis Braille, the inventor of the Braille writing system. He was blind himself and wanted a way to be able to read and communicate, and he developed the Braille writing system. So obviously a huge accomplishment. Did not require vision at all to do that. We've got Stevie Wonder. I've got a few facts here I'm reading. So musician, singer, songwriter. Last I checked, he had 22 Grammy Awards. Didn't need vision to win those. He's an activist. He was important in making Martin Luther King Jr.'s birthday a holiday in the U.S., and he's one of the United Nations messengers of peace. And of course, vision was not needed for any of those things. Something a little more kind of kinesiology related, we have Marla Runyon. In 1994, she received a master's degree, and she did that without vision. Now, her condition is a little different than Stevie Wonder and Louis Braille. She has macular degeneration. So what happened to her is that her central vision, when she got older, and probably in her teen years, she lost kind of her central visual field. So the central visual field, and we'll talk more about this, is important to see things clearly. So think if you're reading a book and you want to read text, you look right at the text. You don't look to the side and try to read it in your periphery, because our peripheral vision is not very good at seeing small details. So slowly her central visual field degenerated, leaving her legally blind, but she did still have her peripheral visual field, which is not good at seeing color, it's not good at seeing detail, but it's actually better than the central visual field at seeing motion. So legally blind, but not completely blind. So she got her master's degree, then she won 5 gold medals in the Paralympics, and in the 2000 Sydney Olympics she competed both in the Paralympics and the Olympics. She was the first legally blind athlete to do that. And she did very well, despite being blind. She finished 8th in the 1500 meters. And I was like, okay, well, how fast did she run, out of curiosity? She had a time of 4:08. The world record, last I checked, was 3:50. So, very good. And you can see her running here. The reason she doesn't have a partner, sometimes like if you're completely blind, you'll have a partner who runs beside you, so that through touch they can keep you on the course. But with her intact vision in her periphery, that must have been enough for her to see competitors and stay on the track. Interestingly, I wanted to see, okay, well, who has the Texas Tech record for the 1500 meters? And that's Sally Kipyego, and her record was a time of 4:07, which is incredibly fast. That was in 2008. She won 9 NCAA championships in 2.5 years here at Tech, and she was so good that after 2.5 years she left Tech and became a professional runner. So a few stories there, but just to say that vision, often a dominant sense, but you don't have to have vision. So for a little bit of an introduction on vision, I found this really neat video about seeing motorcycles. And it's really framed to be kind of safety for motorcycle riders on what to do and what to look out for, because you're not as visible as a car, but they bring up a lot of really spot-on visual science. So give me a pause and check out this video and then come back. Vision is a huge field of research, and one thing we're not going to focus on is the cellular function of vision. So within our retina, there is a chemical translation that occurs when the rays of light hit the retina. That's the photosensitive layer of cells. And eventually, after some cellular processes, there are these rods and cones. And the cones are what allow us to see colors. There's 3 types of cones. We'll talk more about this when we get to color blindness. And the rods, they perceive information in black and white, and they're better at detecting motion. So there's a lot going on here, and we're going to kind of skip over that. What we're going to focus on are the cortical and subcortical visual pathways. And they're depicted here. In this figure, there's a star on it because this is kind of vital for definitely vital for the first half of this module, and we'll see it in the second half as well. So imagine you're standing looking straight ahead at a room, and that room, for whatever reason, has been painted red in the left visual field and green on the right. So those red and green paint colors, light is going to hit them, bounce off, and then hit your retina. And we've kind of depicted that here with these red lines from the red side of the wall and the green lines from the green side of the wall. Now the left and the right eye can see the left and the right side of the world. So if I close my right eye, my left eye, I can see to the left and I can see to the right. Now it is true that my left eye can see further left than my right eye, but we won't worry about that in this course. We'll just kind of think about kind of central, moderate vision where both eyes see both sides of the world. So the information from the left visual field does hit both retina, and we've kind of color-coded where the left visual field hits the left retina and the right retina, and then the same thing for the right visual information hitting, again, both retina. So the first stage here is the retina, and we just kind of said, yes, there's this chemical translation where that's turned from photons or rays of light into action potentials. And we won't worry about the chemical processes. Okay, so from there, we then have the optic nerve. So the optic nerve carries those action potentials from the retina into our central nervous system. So they come in here, and the next structure that they encounter is called the optic chiasm. Now chiasm means X, and it's a fairly X-like looking structure. We'll see some neuroanatomical images later, and it's kind of clearly this X structure. Now the optic chiasm is important because this is where visual information gets organized. So after the optic chiasm, everything we see in the right visual field will now be on the left side of the brain. So note that those green rays of light are now in our left hemisphere. And the red rays of light from the left visual field are now on the right hemisphere. And in an earlier module, we talked about how input crosses, right? Visual input from the left goes to the right hemisphere. Well, that is achieved by the optic chiasm. So now that everything's organized, the next structure after the optic chiasm is called the optic tract. And that carries the information further, and there's an important division here. So most of the information continues on to the lateral geniculate nucleus. And there's a nucleus in the left hemisphere and one in the right hemisphere. That is our cortical visual pathway. What branches off from that, and that's shown in the dotted lines, is the start of our subcortical visual pathway. So this information goes down the optic tract, but instead of going to the lateral geniculate nucleus, it branches off down to the superior colliculus. And again, there's one of these in each hemisphere. This is considered the subcortical pathway because the superior colliculus is in the brainstem. It's not the cortex, it's the subcortex. From there, the information continues on to the pulvinar nucleus, again, one in each hemisphere. And we'll kind of end that pathway at that point, but we will see later on it does continue further. So just to review, the important branching we have here is, you can think of the solid lines as our cortical visual pathway, and just before the lateral geniculate nucleus, we see a branch coming off of the cortical visual pathway, and we call that the subcortical visual pathway, going to the superior colliculi, and then the pulvinar nuclei. Let's continue with the cortical visual pathway, and that's going to be the one we really focus on first. So from the lateral geniculate nucleus, the information travels down the optic radiations to the primary visual cortex. And the primary visual cortex is, right at the back of the occipital lobe. Now, there's not a lot actually on like the surface of the occipital lobe. It actually travels deep into this fissure. So there's just a little bit kind of at the back, but there's a lot of processing that occurs deeper inside the cortex. So that is the cortical and subcortical visual pathways, kind of their neuroanatomy. And now we'll look at a few figures just to see this pathway in different perspectives, different angles. But this will kind of be the key one that we always come back to. So here is, I guess, a drawing of a brain from the bottom, and the hemispheres have kind of been spread out a little. And what you can see is that there's the optic chiasm. So quite obvious X structure. These would be the optic nerves. There's our retina. Don't know why the eyes are cut in half. And then off of the optic chiasm, we have our optic tracts here, just the start of them. Here we're looking at our cranial nerves. So there are 12 cranial nerves. Just double check there for a second. And hopefully you've learned some about these in neuroanatomy, but we definitely don't need to know all of them here. We're focusing on the optic nerve, which is cranial nerve number 2. So here you can see this I guess they cut the cranial nerve, but that would have the optic nerve would have gone to the eye, and then here it comes in, and there's our optic chiasm, and maybe that's even the start of the optic tract. So the optic nerve is cranial nerve 2. Cranial nerve 1 is the olfactory nerve, so that's our sense of smell here, coming out to the nose. Here's a view of the brain stem, and if we look at the back of the brain stem, let's see, we have a good view here. Okay, we'll have a better view later. So what we can see here is this is just the very end of the optic nerve going to the optic chiasm, and this gives us a better view of kind of how the optic tract wraps around here. Okay, here's a better view of the back of the brainstem. So there's kind of these 5 bumps. There's one in the middle, that's the pineal gland, and then the 2 in the top, those are the superior colliculi, and that's part of the visual pathway. Below here, these are the inferior colliculi, and that's not part of the visual pathway. Here's an actual cadaver brain. And again, we see just the end of the optic nerves, the optic chiasm, the optic tract, and you can actually tell here how it wraps around the brainstem. Some of the other structures are, buried deep in here, but you see this giant band of fibers? Those are the optic radiations. And it's neat to see here how kind of large that is, because from the initial image, everything just looks like the same size line, but here you can see those optic radiations are quite huge. Let's see, the lateral geniculate nucleus, they're saying it's right here, although that's not something that I'd be able to landmark from this image. Here we're seeing an image of the primary visual cortex, the destination of the cortical visual pathway, and this shows again nicely that there's just a little bit on the surface, but then there's this fissure here, and there's a ton more processing within the cortex. Now, one way you may have heard the primary visual cortex referred to before as Brodmann area 17, and there's some interesting history behind this. So Brodmann was a German anatomist, as full name Corbinian Brodmann, kind of a cool name, and in 1909 he published kind of what we're seeing here, this research where he identified different regions of the brain. And the way he did this, he didn't have fancy technology like fMRI that we have nowadays, but what he did is he looked at the cadaver brains with microscopes and just started in one area. Now, area 1 is right here, so that's maybe where he started. And then he would move in a different direction, and every time the cellular architecture of the brain changed, he labeled that a new area. So you can see we've got 1, 2, 3 in here, and then if we go forward, we've got area 4. Let's see, where's 5? Okay, 5 is hidden back here. We've got area 6. So, eventually he came down to the primary visual cortex, and he called this, area 17. So the fascinating thing about this is he had a microscope, basically, and tools for staining cells, but he didn't know that area 17 was the primary visual cortex. He could just see that the cellular architecture was different than all the other areas. What we now know is that, well, when the cellular architecture of the brain changes, the function of that area of the brain changes. So even though this research is from 1909, over 100 years old, we still use these areas because they tell us even more than just cellular architecture, but also function of the brain. So eventually we realize, well, that Brahman area 17, what does it do? That's our primary visual cortex. Another name for Brahman area 17 is the calcarine fissure. So the fissure, this kind of gap where we see the primary visual cortex, is named calcarine, and also sometimes called the striate cortex. But we'll probably stick with primary visual cortex. Here's our last kind of novel view of the visual pathway. We don't see the entire pathway here, but we see our eye, and the retina should be somewhere in here, and the optic chiasm is kind of lost, but we've got our optic tract, we've got our lateral geniculate nucleus, and you can see those are those giant optic radiations that go back to the primary visual cortex. Okay, so that's kind of a review of our neuroanatomy. To reinforce our understanding of that, let's look at what happens when we damage different parts of the cortical visual pathway. So we'll just stick to the cortical pathway, the solid lines. We won't talk about damage to the subcortical pathway at this point. So in terms of visual damage, there's a few words we need to know. So the first is anopia, and that just means absence of sight. And we're going to look at more specific examples of anopia. So there's hemianopia. This is loss of vision in half of the visual field. So if your left eye, your right eye, they only see one side of the world, that is an example of hemianopia. There are multiple types of hemianopia, and we'll see those later. Quadrantanopia, kind of a well-named term, is loss of vision of a quarter of the visual field. And scotoma, this is loss of vision in a part of the visual field, also known as a blind spot. And hemianopia and quadrantanopia are in bold here because we'll see more examples of these next. So we've got a slightly different view here of the cortical visual pathway, kind of just flattened out, if you will. And what we're going to understand and learn is, well, what happens if we damage area 1? So if we damage this area, what happens to our visual experience? Then we'll look at 2, 3, 4, 5, 6, and 7. So we've got 7 different ways we can damage our cortical visual pathway, and we'll see how that impacts our perception. Some of these pathways can occur from specific conditions, and when we look at example of those, I'll bring those up. Others are maybe more theoretical. So number 2, for example, well, it would be weird to somehow damage just this side and just that side. So 2 is kind of really the opposite of 3, and 3 is a condition that can occur, but we'll get to that. All right, so how are we going to do this? So let's think of our vision like this. So this is our left eye, and this is our right eye, and the left eye can see the left side of the world and the right side of the world, and the right eye can see the left side of the world and the right side of the world. We're not going to worry about kind of the extreme peripheries where only one eye can see that far left and only one eye can see that far right. We're going to just stick with our mid-visual field. So this is how we'll depict normal vision. Now if you have no vision at all, so imagine you cover both your eyes, the way we'll depict no vision at all is we'll shade in all 4 of these areas. What we're going to see in 1 to 7 though is something between full vision and no vision. We're going to have these selective losses in vision, specifically examples of hemianopia and quadrantinopia. All right, let's look at number one. So number one, the situation here is your left optic nerve has been cut. This could also be, you've lost or damaged your left eye. That would also have the same impact. And one is kind of easy to think about because we can all try it. So we can all just close our left eye, and then that is what this visual experience is like. So, one is not too hard to appreciate. So if we kind of draw what would it look like, well, if I close my left eye, the right eye sees everything, and now my left eye can't see the left side of the world, and it can't see the right side of the world. When we get to 2, 3, and on, we can't really try these, so we need a better way to identify what visual information has been lost. And we're going to try that with number one, even though we already know what it would be like, but we're going to confirm this technique will work, and we're going to rely on this for the subsequent ones. So let's go to number one here, and we have cut this red pathway and this blue pathway. And you can think of these as pipes or wires, and if we cut those pathways, electricity no longer flows, or the water will no longer make it to its destination. And I guess the destination here would be the primary visual cortex. All right, so we've cut number one, and instead of tracking it back to the primary visual cortex, what we want to do, or I guess maybe I should say tracking it forward to the primary visual cortex, we want to go back to see where this visual information came from. So one here, well, let's trace it back. Okay, we've made it to the left eye, so we're going to lose something from our left eye. So to note that, I'm going to circle this yellow. And then which side of the world it is, well, we got to travel back on these arrows to see which side of the world it is. Oh, and it's a blue line, so it's the left side of the world. So from this cut, just half of the cut on this side, we're losing the left visual field in the left eye. So I'm going to shade that in gray. That's only half the damage. We've also cut this pathway, so we're going to do the same thing. We're going to trace it back to the eye. Oh, we get to the left eye. So we're going to lose something in our left eye, and now it's actually the right side of the world. So we're going to shade that in. So with both techniques, kind of thinking about it conceptually, which is easy, we can close an eye, or tracing it back, we see that the left eye has lost vision, and the right eye can still see the left side of the world and the right side of the world. But this tracing back method is going to be helpful in the subsequent examples. Oh, when would this happen? Well, it's possible to damage your eye, poke your eye out, something along those lines. The other somewhat common way is with multiple sclerosis. That condition can attack the myelin sheaths of any neurons, and it tends to be more common that it attacks the myelin sheath of the optic nerve. Now, it could do it on both eyes, and then you'd have visual deficit in both eyes, but it can also be one eye, and if there's enough of the optic nerve that becomes demyelinated, the action potentials won't make it further down the pathway, and you could lose full vision in your left eye. Okay, let's look at number 2. So we're going to do the tracing back technique again. So 2, we've cut just this red pathway and just this blue pathway. So let's trace the blue first. This goes to the right eye. So we're going to lose something in the right eye, and it's the left side of the world. So we got to go to the right eye, left side of the world, and we're going to shade that in. The other half of the damage is here. So we're going to trace this red pathway. It's going to go to our left eye, and it allows our left eye to see the right side of the world. So left eye, right side of the world, we're going to shade that in. So what does this look like? So this is our first example of hemianopia, because both eyes have lost half of the world. The specific type of hemianopia is called binasal hemianopia. So it's specifically telling us which half of the visual field has been lost. So bi means that it's both eyes, And nasal means that it's the inner part of the visual field. So think, where's your nose? We've lost the inner part of both eyes, as shown here. Now this type of hemianopia, with both of your eyes open, your right eye sees the right side of the world, your left eye sees the left side of the world, that would make it to your brain, and your brain would stitch those two images, and your full visual field with both eyes open would be largely intact. If you close one eye, then all of a sudden you would lose half of the visual field. So if I had to pick what type of hemianopia I had to have, binasal hemianopia is one of the better options, because with both eyes open, your brain would stitch together those two half images into a whole image. Let's try number 3. And here I'm going to try tracing it on myself. So let me just change to a pen, and let's change this color. Let's go with yellow like before. Okay, so number 3, these pathways would be preserved, but we've cut these two in the middle. So maybe let's follow this one first. So we're going to go to the left eye, and this allows the left eye to see the left side of the world. So we're going to need to shade this in. I guess that should be great, but I think PowerPoint will fix that for us on the next slide. Okay, it's not a perfect shading, but hopefully you get the idea. We've also cut this pathway. This is going to go to our right eye, and it allows us to see the right side of the world. So we're going to shade this in. So notice that 2 and 3 are opposites of each other. And that makes sense, because 3, we've cut these two pathways, 2 are the opposite. So we really should have opposites in what's being lost in number 2 and 3. Okay, there we go. That's better filling in than I did. So what would 3 look like? Let's take a look. Oh, before we see it in more detail, when could this happen? This can happen when you have a tumor on your pituitary. So here's our pituitary, and right above it is the optic chiasm. Now surrounding the pituitary is bone. So if you get a tumor on this structure, a growth, and it grows big enough, it's going to take up this cavity, and then once it's full, there's going to be nowhere for it to go but up. And it can end up pushing on the optic chiasm, but at first just the middle of the optic chiasm. And what you can end up is with a loss of the left eye, the left visual field, the right eye, the right visual field. This more specifically is hemianopia, and more specifically bitemporal hemianopia. So we've lost it in both visual fields. Temporal, so think of your temporal lobes, we've lost vision on both sides. And with with both eyes open, this wouldn't be the worst type of hemianopia, because the two halves of the world that you do see, your brain would stitch together. The biggest problem is that the left eye, no eye can see very far left, and no eye can see very far right. So you would lose those extreme peripheries, but your vision in the mid-visual field would be intact. But you've lost the extreme left and extreme right, even when both eyes are open. Let's take a look at number 4. So number 4, we've cut the 2 red pathways here, and that should already hint at what we're going to see. But let's trace this back. So this one goes to the left eye, and it allows us to see the right side of the world. So left eye, right side of the world, and PowerPoint will fix that in a moment. And then we've lost this pathway, so this is going to go to, off a little bit here, off to the right eye, and it allows the right eye to see the right side of the world. So here we've lost that. So number 4, we've cut those red pathways, so both eyes are going to have to lose, the right side of the world. So this is also known as right homonymous hemianopia. So right means we're losing the right side. Homonymous means we're going to lose the same thing in both eyes. And hemianopia, we've a loss of half of the visual field. And with this type of hemianopia, even with both eyes open, you're only going to be able to see the left side of the world. So we have 5, 6, and 7. And for 5 and 6, life gets a little more complicated. So here we have the lateral geniculate nucleus, and there's a very complex mapping that occurs in this nucleus. It's not as simple as it looks. It's not just that this pathway goes here and that pathway goes here. So this pathway connects to this side, and it also connects to that side, and the same thing with this this part of our optic radiations. They connect to both sides here of the optic tract. So it's not as simple as tracing it back. What we need to do is just remember that 5 and 6 are going to be similar to 4, but they won't be as bad. For 5, the more lateral parts of the optic radiations provide vision for the upper quarter of the visual field. And 6, the more medial aspects of the optic radiations provide vision for the lower quarter of the visual field. So for 5 and 6, what I like to remember is that they're like 4, but not as bad. 5, we're losing the top quarter. So this is our first example of quadrantinopia. And this is what that visual experience might look like. The previous images came from Wikipedia. They didn't have one for quadrantinopia, so I took out a quarter of the visual field here, but it wouldn't be so abrupt. There'd be this quick transition from full vision to no vision. This is also referred to as right, homonymous, superior quadrantinopia. So right, we're talking about the right side. Homonymous, same thing in both eyes. Superior, it's the upper right quarter, not the lower right quarter. For 6, it's going to be like 4, and the opposite of 5, it's going to be the lower quarter. And back to 7, although we can't really trace it back through 5 and 6, 7 we can think about, it's got to be the same as 4, because we've lost both red visual pathways. So we're back to this very classic hemianopia, where both eyes cannot see the right side of the world. That was a lot on damaging the cortical visual pathway. So if you want now, you can tackle the first half of the practice questions. They'll go over those same 4 examples. If you can try doing them on your own, make sure it makes sense. And then there's a kind of a mirror image where the damage is now, instead of on the left hemisphere, it's in the right hemisphere. And you should be able to use the same techniques to figure out what vision has been lost in all of those examples. So, if we think about the cortical visual pathway, we start at the retina and eventually make it to our primary visual cortex. Now, the primary visual cortex is not where vision ends. So once we get there, yes, it allows us to see some basic features of vision, some very simple things, but there's a ton more visual processing that occurs in the occipital lobe, up in the parietal lobe, and even the temporal lobe. So visual processing is distributed throughout a lot of the cortex. And what we're going to take a look at now is some of these higher visual areas and what function they perform. And the brain kind of has a strategy where it divides and conquers. So it says, okay, area V5, you focus on this. Area V2, well, you focus on this. So there are some examples, and we'll look at those, where a single area does a single visual function. Other ones can be more broad in their visual processing abilities. The other thing about visual processing is it's not simply a sequential series of stages. So if we look at V1, so yes, information does go from V1 to V2 to V3 to V4 to V5, so that's sequential. But area V2 will also send information back to V1. And if we look at V1, so it will send information to V2, but it also sends information directly to V5. So we also have parallel processing occurring. So our visual processing system is very complicated. And even this is a simplification. This is a slightly more exact diagram of the visual processing areas and how they're interconnected I think this is actually from a monkey. which they have very similar visual processing areas that we do. So we won't worry kind of too much about the super complexity here, but we will look at a few areas that do very specific things, functions of our visual processing. There's one more example if you want to try reinforcing your knowledge of loss, damage to the conscious visual processing. This is one that I often do in class. And there's just 3 here, but these are, well, I guess you've probably already done this one, but example X and Z here are a little bit different than the other ones because instead of, well, I guess we've already seen this one here. Okay, we're going to have to go with X as the only unique one. So if you did the sample questions, you've already done Y and Z, but X is a little bit different. It's not the opposite of one. We're saying, okay, we've just lost this one. So if you want a couple more questions, you can tackle those or save them for practice later. Oh, there are the answers. Okay, so let's get to those areas that do specific things for visual processing. And we're going to look at 4 different conditions: akinetopsia, achromatopsia, prosopagnosia, and blindsight. And I find these are each kind of fascinating in their own way. First off, visual area V5. It is dedicated to allowing us to see visual motion. So no other parts of the brain allow us to see visual motion. That's specifically the function of area V5. If you damage that area, the condition is called akinetopsia. So A means without, kin, so think kinesiology, movement, and opsia is a visual disorder. So akinetopsia, think about a visual disorder where you can't see movement. This is also called motion blindness. This is very rare because you have an area V5 in your left hemisphere and your right hemisphere. And they found that if you lose only one, the other one will take over and you still see visual motion normally. So you have to damage area V5 in both hemispheres. Now I have a quick little story here about an individual with akinetopsia. So it's hard for us to understand what it must be like to see but not be able to see motion. So the visual disorder complained of by the patient was a loss of movement vision in all 3 dimensions. She had difficulty, for example, in pouring tea or coffee into a cup because the fluid appeared to be frozen like a glacier. So she can see the water, but it's just these still images. In addition, she could not stop pouring at the right time since she was unable to perceive the movement in the cup or pot when the fluid rose. Furthermore, the patient complained of difficulties in following a dialogue because she could not see the movement of the face and especially the mouth of the speaker. In a room where more than 2 other people were walking, she felt very insecure and unwell, and usually left the room immediately because people were suddenly here or there, but I have not seen them moving. The patient experienced the same problem but to an even more marked extent in crowded streets or places, which she therefore avoided as much as possible. She could not cross the street because of her inability to judge the speed of a car, but she could identify the car itself without difficulty. When I'm looking at the car first, it seems far away, but then when I want to cross the road, suddenly the car is very near. She gradually learned to estimate the distance of moving vehicles by means of the sound becoming louder. So it must be a very strange condition. I have a picture of a strobe light here because that's maybe our closest experience. With this strobe light, you get these static images, but they come at a set interval, and our brain kind of still turns them into motion, although the motion is different than normal motion. That's not the case with akinetopsia. Those images don't necessarily appear in a set pattern. So when she's looking at 2 people, she'll see one view of them, and then the next time she gets a fresh image, might be 1 second later, it might be 2 seconds later. And even if they were coming in at the same pace, the part of her brain that would turn that into motion, like when we see someone, say, dancing with a strobe light, well, that's V5, and for her, it's broken. But that's maybe the closest parallel that we can get to. Area V4 is dedicated to seeing color. If you damage area V4, the condition is called achromatopsia. So again, A without, chroma color, and opsia is a visual disorder. This is also referred to in a few other ways. Sometimes it's called complete color blindness or cerebral color blindness, to know that the deficit is in the cortex and not in the retina. Now this is very rare. Far more common is partial color blindness. And partial color blindness is so much more common that sometimes people say, oh, I'm colorblind, and what they're meaning is that they're partially colorblind. It's just complete color blindness is so rare that we've kind of co-opted the term color blindness to typically mean partial color blindness. So before we get more into complete color blindness, let's take an aside into partial color blindness. It's fascinating in of itself. So partial color blindness is typically caused by a deficit or inactivity in one of three of the cone cells. So we have three cone cells in our retina, and they allow us to see red, green, and blue. And if you're looking at a color that's yellow, for example, then it would activate two cone cells, and that would be interpreted as yellow. But if you're missing a cone cell or two types of cone cells, then you have partial color blindness. So there are neat tests for partial color blindness. I thought we'd take a quick look at them. Now, one thing about partial color blindness is that it's not uncommon, or I guess I should say it's somewhat common. And it's most common in individuals of Northern European ancestry. So of those individuals, 8% of males are partially colorblind, but only 0.5% of females. And the most common type of color blindness, because depending on which cone cell you lose, you'll have difficulty distinguishing different colors, is red-green color blindness. And students in the past, I've had a few who've mentioned, oh yes, I actually am partially colorblind. So let's look at these tests. This isn't a clinical assessment, but we can see if perhaps you are partially colorblind. So these are the Ishihara color plates. So they're these round diagrams, and what you're trying to do is find a number within them. So this first example is a control, and everyone hopefully can see this 12. Even if you are totally colorblind, you should be able to see the 12 within this plate. So I'll show the plate, and then we'll kind of drop the background for the more complex ones where you may or may not be able to see the numbers. Here's our second one, and I'll drop the background. But with the background on, if you saw only a 3, that could indicate that you have red-green color blindness. There are different types of red-green color blindness, so it gets a bit complicated. Now, if you are totally colorblind, you wouldn't be able to see the number 8 at all until we drop the background. Next example. So see if you can identify the 2 digits here. Now this one, most colorblind people cannot see this clearly, but without the background, you can see that what was hiding there was a 73. And this one I enjoy. So before you panic, the good news is if you have all 3 cone cells, you won't see a number. And if you are red-green colorblind, you actually have an advantage. You should be able to see a 5. Now I have 3 working cone cells, so I can't see the 5. If you are red-green colorblind, you can see the 5. Send me an email and maybe draw on where the 5 is, because I'm quite curious about this one. So what I like about this is it shows that red-green color blindness or partial color blindness isn't necessarily bad or worse. It's just different, and in this case, it's actually better. Okay, and this one, we'll drop the background. So there's a 2 and a 6 hiding in there, and this one's neat because if you have red color blind or mild green color blind, you would only see the 6. But if you're green colorblind or mild red colorblind, then you would only see the 2. Not quite sure if I understand that. But this one can distinguish different types of color blindness. All right, So that's partial color blindness, typically caused by cone cells. Let's get back to complete color blindness or cerebral color blindness caused by damage to area V4. Oliver Sacks, a famous neuropsychologist, a medical doctor who wrote many different books about many different fascinating conditions, and he's got a whole book about being colorblind. We'll watch a little video that was created after the book was released. And in this book, he talks a lot about this island Pingelap. So it's so small, it's not even an island, it's classified as an atoll, and it's in the Pacific Ocean, and it's only 1.8 kilometers squared, so fairly small. And there were 250 people on this island with its own language, its own culture, somewhat similar to other Pacific cultures, but uniquely its own. And what happened is in 1775, there was a very bad typhoon, and it killed 90% of the population, leaving about 20 people. And what must have happened was one of those survivors was a carrier for the colorblind gene. So they weren't colorblind themselves, but they were a carrier. And this would be the type of colorblind where they're completely colorblind, they don't see any colors. So that's very rare in the world, but it became common on Pingelap. So 4 generations after the typhoon, 3% of the population was colorblind. 6 generations later, 5% were colorblind. And now 10% are colorblind and 30% are genetic carriers. So it's this fascinating place where being completely colorblind is common. It's not the majority of people, but in other in the world, if you're completely colorblind, you might never meet someone else who's completely colorblind. And here, likely your neighbor or someone in that household is probably completely colorblind. So there's a YouTube series on this, and you can just watch the first video about Oliver Sacks kind of connecting with a Norwegian gentleman who's completely colorblind, and then they go to this island to see what life is like. And they have some interesting conversations about, seeing color, not seeing color, and what their different and shared experiences are like. So pause this and go watch just the first part, the first video in this series of I think 6, and then come back here. For our third condition, we're going to look at the fusiform gyrus, and it's located on the bottom of the brain. So we've got the brain flipped over here so we can see the bottom. And you have a fusiform gyrus in the right hemisphere and the left hemisphere, and we're going to focus on the one in the right hemisphere. It allows us to identify faces. And if you damage that area, the condition is called prosopagnosia. So proso means face, and agnosia means without knowledge. So you have vision, but without knowledge of faces. And this must be very bizarre. Oliver Sacks, in one of his books, wrote a story about an individual who had prosopagnosia. So you call him the man who mistook his wife for a hat. He also had other visual disorders, so it was kind of even more fantastical. But what I have here is an example from that Oliver Sacks book of a patient who had exclusively prosopagnosia. So it'll give us a better idea of what this symptom is like. Their patient was a young man of 32 who, following a severe automobile accident with unconsciousness for 3 weeks, so serious brain injury, complained exclusively of an inability to recognize faces, even those of his wife and children. Not a single face was familiar to him, but there were 3 he could identify. These were workmates, one with an eye-blinking tic, one with a large mole on his cheek, and a third because he was so tall and thin that no one else looked like him. Each of these was recognized solely by a single prominent feature mentioned. In general, he recognized familiars only by their voices. So those 3 people, it's not that he can, their face recognition is preserved. No, he's identifying very specific things about them that, are completely unique to those individuals. He had difficulty even recognizing himself in a mirror, which must be very strange. In the early convalescent phase, he frequently, especially when shaving, questioned whether the face gazing at him was really his own, and even though he knew it could physically be none other, on several occasions grimaced or stuck out his tongue just to make sure. By carefully studying his face in the mirror, he slowly began to recognize it, but not in a flash, as in the past. He relied on the hair and facial outline and on 2 small moles on his left cheek. So when he sees his face, or imagine when you see your own face in a mirror, you immediately recognize that's my face. And the part of the brain doing that is your right fusiform gyrus. Without that, you can see your face. He's not blind to faces, even though this is sometimes called face blindness. You see the face, you see all the individual features, but then your brain putting that together and saying, oh, that's my face, or oh, that's my friend's face, that part is gone. It was also evident that visual memories of people, even from long before the accident, were severely impaired. There is memory of conduct, or perhaps a mannerism, but not of visual appearance of face. And that's because if you want to think of a memory of a face, that memory comes out of your long-term memory, and it gets replayed, if you will, in the same areas of the brain that you used initially to recognize the face. So although that memory probably still has face information, to see that, you need your fusiform gyrus, which is damaged. So you wouldn't have memories of faces, you also wouldn't have dreams where you could recognize faces. A final humorous point. He was also unable to recognize his wife, needing her to identify herself by a visual marker, by a conspicuous article of clothing such as a large hat. So a few more things about face perception in the fusiform gyrus. So let's go back here for a second. So for this example, it's critical to remember that it's the right fusiform gyrus that identifies faces. The left fusiform gyrus is used to detect kind of everything else besides faces. So I was watching this Bond film a few years ago, and I was quite tickled when the villain decided that he wanted to give Bond prosopagnosia, which is a very odd torture technique. So if you want to just watch this quick video clip, it'll start kind of in the middle, and look at which side of the brain the villain mentions that he's going to damage to give Bond prosopagnosia, and see if it makes sense from what we've learned. So this is the bizarre thing. I had to kind of double check when I watched this, but it said here, the needle's going to go right in here. So that's, the left side of Bond's face, and the problem is face identification is the right side. So he got it wrong, and maybe that's why in the film, even though the needle went in, he could still recognize Madeline. I think that was her name. Yep, Madeline or Madeline. I think she's French, right? So I'm not sure if that was intentional or a bit of a faux pas by the filmmakers. Another thing about the fusiform gyrus is it's good at detecting upright faces. And here we have some upside down faces, and they still look like faces, but they confuse the fusiform gyrus because we're not used to seeing upside down faces. And in this example, the question is typically, which face is more attractive? It's like, well, aren't they the same? Are they, equally attractive? Let's rotate these to be upright, and there'll be no trickery. They're just going to become upright so the fusiform gyrus can properly identify the faces. So now that they're upright, and if you're asked which one is more attractive, it should pop out that something is weird here. And what has been done is the eyes are flipped upside down, and the lips are flipped upside down. I think everything else is the same. But when that was upside down, even when you know that something's wrong, it doesn't look as weird. And that's because the fusiform gyrus is best at detecting upright faces. We're not so good at putting facial features together into a face when they're upside down. So one thing I've kind of mentioned in passing is that the fusiform gyrus allows us to see faces. We can still see all the features of a face without the fusiform gyrus, but it's putting that together and recognizing who it is that we need the fusiform gyrus for. Now, if I showed you a single feature of someone's face, even if it's someone famous or very familiar to you, you probably won't know who it is. Now, to give you an advantage here, I've picked some eyes of people who have very, very famous eyes. So it's possible that just with these eyes, you recognize who it is. But typically, to recognize someone, we need our fusiform gyrus, and we needs to see multiple features of the face. Not necessarily all, but more than just a single one. So does anyone recognize who this is? I tried to go with someone who might be more, you might know more about than, say, a 40-year-old celebrity that I might know. So this is Bella Hadid, who I think is famous for her eyes. And if you didn't get that one, maybe you'll get this one. This is a male celebrity, also famous for his eyes. I'm trying to think. I feel like there's fewer male celebrities known for their eyes. Not sure. But this one is Cillian Murphy. Or is that pronounced Killian? Whew, I'm not sure. One last thing about the fusiform gyrus for face perception. It's a bit overactive. And it seems that we've evolved in a way that we'd rather misidentify a face, so see something and be like, I think that's a face when it's not, than miss a face in a crowd. And that could be important in a culture where you need to be able to pick out, friend and foe and enemy, and not miss someone maybe hiding behind a bush who wants to jump out and kill you. But it leads to kind of these funny things where we often see faces in inanimate objects. These light switches look very mischievous. This exercise equipment looks mad at you. Maybe you haven't worked out in a long time. And I always enjoy this one. So you have a smile, it's on the house, quite literally. So we've got one more example to look at, and I think this is the most fascinating one from all of today. We saved the best for last here. And what we're going to look at is, well, what happens if you lose your cortical visual pathway? So we're going to look at damage to the primary visual cortex. So imagine you have an accident, tumor, seizure, sorry, not seizure, stroke, something that permanently damages your primary visual cortex. So information still flows from the retina, but it doesn't get here. And if it doesn't get to V1, it's not going to make it to V4, V5 for color and motion. It's not going to make it to the fusiform gyrus to identify faces. You've lost all of your cortical visual processing. What is intact is your subcortical root. So if just this area is damaged, then visual information can still make it down to the superior colliculi and then up to the pulvinar nuclei. And this part of our visual pathway, the subcortical pathway, does do some very important visual processing. It's not the dominant pathway. So this pathway is about 90% of the visual processing, but 10% of the axons related to visual processing are in the subcortical pathway. So 10% is a lot less than 90, but this 10% is still more than all the axons involved in auditory processing. So it's still a significant part of the brain. So let's see. How can we learn what the subcortical visual pathway allows us to do? And first what we're going to look at is a monkey who had its primary visual cortex removed. So we've got another second kind of simplified diagram here for visual processing. We'll see this in this module and the next. So we have information, visual information reaches the retina. We've got this divide. We've got a cortical pathway. So we're not labeling all the tracks, but it goes from the lateral geniculate nucleus to the primary visual cortex. And this monkey had its primary visual cortex completely removed. So we know nothing is going to make it to V4, V5, any other cortical visual area. But it does have a preserved subcortical visual pathway. From the retina, it goes to the superior colliculus, and then the pulvinar nucleus. So the monkey had this surgery, it recovered, and then they put it through a series of tests to see what its visual processing was like. So give me a pause, check out this video, and come back. So the monkey's name was Helen, and in that video, the strange thing is it looks like Helen can see. So they put her into this observation area, and she had done this before where they put out obstacles, and she successfully navigates around them. Now, if you were blind, you would run into them, right? Or you'd at least move slowly to see what's in front of you. But Helen moves around like she can see. In the last part of the video, she's sitting at a desk and they're throwing food down, and she moves her head to where the food lands, picks it up, and eats it. You know, she's not behaving like she's blind at all, which is very weird. She's got no cortical visual processing. So this is strange. Now, the challenge with Helen is that we can't ask her, hey, Helen, what do you see? We can just do these sorts of tests and try to interpret the results. So let's look at humans instead. So this is an individual also with a completely damaged prior visual cortex. Now, they wouldn't have surgery to do this on purpose because that's unethical, and perhaps even so with animals, but animal research and the ethics behind it is constantly changing. So the study with Helen, I'm not sure if you could even do that sort of thing nowadays. Anyways, let's see what's going on with this individual. So behind him is his doctor there just in case, and they brought him to this hallway, and they start by saying, hey, what do you see? He says, I don't see anything. And they say, okay, well, what do you mean? He says, well, I'm blind. And, well, explain. He's like, well, before my accident, I could see things, and now I'm blind. I see nothing. And he's not lying. That's true. He is blind. And that sounds different than Helen, right? He's telling us he can't see, but we couldn't ask Helen. So the doctor says, okay, you're blind. We brought you to a hallway, and in the hallway there's obstacles. Can you see those? No. Okay, well, I just want you to walk straight to the end of the hallway. You know, I've pointed you in the right direction. Now move ahead. So give me a pause, watch this video, and come back. So the fascinating thing here is now this individual with blindsight behaves very similarly to Helen. He didn't run into the obstacles. Right? When the garbage can came up, he turned to the side. He successfully navigated all of them. And if the doctor asked, well, he's turning and say, hey, why are you doing that? He'd say, I don't know. He said, well, you're avoiding an obstacle. He says, oh, am I? Yeah, what is it? He'd say, I don't know. And he's not lying. So it's this kind of fantastic, amazing condition. Probably not good to have, but very illustrative to us of the difference between the conscious and the cortical and subcortical visual pathways. So the cortical visual pathway is what gives us conscious perception. And that's all we can really think about. When I think about my visual experience, all I'm aware of is my conscious perception. But there are parts of my brain that see things unconsciously. And this is the role of our subcortical visual pathway. It gives us unconscious perception for action. And we call this blindsight. So these individuals, they can't see things, but they can make movements that are appropriate within their environment. So if someone had blindsight, so imagine I'm a patient with blindsight. I'll close my eyes to depict blindsight, although, of course, their eyes would be open. And the doctor holds up something and says, what is this? I says, well, I don't know. I don't see it. And then if my eyes are open and they toss me the ball, my subcortical visual pathway would see it, and it would cause me to react. Maybe I'd put my hand out to catch it. Maybe I would dodge. I would act appropriately using my subcortical visual pathway. Now, because my conscious pathway is broken, that information would never make it to consciousness. So if I had dodged the ball, the doctor would say, well, why did you do that? And I would say, I don't know. But it's like my body responding automatically outside of consciousness. So a fascinating condition. We're going to look at a video in just a moment here of an individual named Graham Young, and he has blindsight, but only in half of his visual field. So he had an injury. He mentions a road injury. I'm not sure if that means it was car or bike, but some sort of road injury, and he permanently damaged his left primary visual cortex. So that's very similar to kind of 7 here. 7 could be the optic radiations, or if maybe we put a little further, that's our primary visual cortex. And if we remember our mapping, he's going to lose his perception in his right visual field. So in his left visual field, he'll still see things consciously, and he can react to things, subconsciously. In his right visual field, that's kind of the fascinating visual field, where we'll show him something, he won't be able to see it, but he will be able to make movements with the use of his subcortical visual pathway. So check out this video on Graham Young, and it'll start in the middle, and just watch to 23, 25, because it will transition to a third topic after that, and we only really need to watch the part about blindsight. So blindsight, fascinating condition. Sounds like an oxymoron, but it's literally seeing what you don't see. This occurs from cortical blindness, so you have damage to your cortical visual pathway, but there needs to be preserved information still traveling down the subcortical visual pathway. Now these individuals, they say basically that they're blind, and that's true because they have no conscious perception. But they can automatically, almost reflexively, if you will, respond accurately on these visual motor tasks. You think of a reflex, like if I do a tendon tap reflex, tap your patellar tendon, your knee kicks out, that is mediated without consciousness. You didn't need to be consciously aware for that tap. You could be asleep, and I could tap your knee, and you should have that same reflex. And it's kind of the same thing here. You have these intact visual motor abilities outside of consciousness via your subcortical visual pathway. Now that subcortical visual pathway, we talked about it stopping at the pulvinar nucleus. In the next module, we'll see that information actually does continue on to the cortex, to the posterior parietal cortex, which is partially responsible for those intact abilities we see in Graham Young and individuals with blindsight. So there's a few more sample questions to kind of test your knowledge of these cortical visual areas. Make sure to study all this content well, and when you're ready, you can tackle the test. Good luck with that, and I'll see you in our next module.